I obviously got in somehow.
Hey, yo, Dip, drag me right quick.
When he gets Twitter. He's very racist. Why is he so good at football? Pure racism.
That doesn't make sense. That doesn't make sense. What, how does that make sense?
Everybody's playing.
Yo, Dip. It doesn't work like
You called them for IT support?
Wait, nobody can log in.
No, that's not true. I've got a bunch of kids in here right now. I have a bunch.
That's a. Some people can log in and some people can't. And Mr. knows
What you want them to do? Ask him his name frame?
Dip says he tried everything.
Hold on a second. There's a lot of kids that are getting in. I'm gonna ask them. Hey, young men and women, I'd like to know, how are you guys getting into Schoology? Please come on your mic and not chat to me. It makes it so much easier.
I was just able to log in like normal.
Do you have it bookmarked or do you have it, are you going through ClassLink?
I have it bookmarked. I don't use ClassLink.
Okay. Is that true for everybody here? You can drop that in the chat. Are you doing it the
Yeah.
same way? You're going through a bookmark?
Yeah, mine has a bookmark as well.
Okay. Everybody here is going to their bookmark. So when you do get in, you need to bookmark it. That way you can go directly to it instead of going through ClassLink. You should be able to use that address I sent through Remind.
What's your mic code?
See, I'm gonna see if you can do it. Try that. I'm gonna wait 5 minutes and then I'm gonna start the lesson. Guys, we're gonna wait until 8:35 and then we're starting the lesson with whomever's here. It is 8:31.
Mr. Pope,
Yes.
one of my friends is asking for the Zoom link. Can you send it through me to the chat so I
Yeah.
can send it to?
Yes, I can. Yes, I can. I'm gonna have to stop sharing this to do it, but yes.
Hey, man, he usually knows what to do.
I need to let a bunch of other kids in real quick. There's a bunch of kids are making it now. Only the ugly kids get it. We'll wait 3 more minutes just to be cool. Let everybody get in. Oh my gosh. Oh, that's great. I may not be able to get in. I can. Do you guys know if I send a meeting ID, whether a meeting ID will work? Because I have to close this call to get the link to share with you guys. Are you guys okay with me?
Oh,
closing?
never mind. I don't need it no more
You don't need it anymore? Why not? But
Yeah.
other people might.
because apparently Diana sent it to him.
Diana sent it?
Yeah.
All right, good. Okay, good. Oh yeah, there's, good. There's 21 of you guys in now, or 20. Most of you guys are getting in. I'm gonna go ahead and put the do now back up. We'll still start at 8:35. We'll be 5 minutes off.
Yeah.
All right, let's get in, let's go. Do you have the Zoom link, Mosadi? You gonna email him the Zoom link?
Yeah.
All right, do it. Let's go, let's go, let's go, let's go, let's go.
What's your email, Jonathan? Here, bro, just put it
This phone.
in. We're all struggling right now, okay? Yes, sir. I made that email when I was 12. Been using it ever since.
Are you proud?
Come on, bro, where you, bro? It's just that you call him HR.
Stop it, bro. This is so tighter. I feel like you're touching something. Hold on.
Why? Why are you worried?
What do you mean, why am I worried? You want me to go pick your boy? Are you worried?
All right,
Bro,
all right, Caleb, you're in the call? Caleb, are you in the call?
Yes.
Okay, do now. All right, we're going to take an answer to the do now in say 30 seconds. You guys are here. I don't like being, I'm sorry that we're off in terms of our time.
Stop it.
On the count of 10, go ahead and put, yeah, go ahead and mute yourself. Drop it into the chat. A, B, C, or D. And you can do it openly or privately, doesn't matter to me. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Drop. Let's see what you got.
I sent it.
I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 correct answers so far, 15 correct answers, 16 correct answers, 17, 18. Okay. Most of you guys see the ones that are being shared. You know, there's some that are being shared with everyone. It is going to be D. In this problem, you never needed to read the problem at all. It's pointless reading the problem. What is important when you looked at the question, it says, which of the following is supported by the data? So you're going to immediately look over here at these ratios, and these are essentially ratios, right? And so if I look at this, it's basically a 5-to-1 ratio for these phenotypes. And when I look at that compared to these, I know that it's going to be immediately, it's a gene-linked scenario. These two genes, these gray and long wings, gray body long wings are linked. Gray black body apteris wings, that just means these apteris wings, these are vestigial wings, that they are linked. They're linked, and so I'm looking for that here. I know that linkage is the same chromosome and very close on to the same loci. Therefore, D is my answer. And it took me, honestly, this question shouldn't have taken you 30 seconds. You're just looking at the ratio and knowing that if you see them like this, that it's got to be linkage. All right, I don't know who can and cannot get to Schoology. I'm going to stop sharing, go to Schoology. We're going to look at what we're going to do today. Hopefully you can. It's going to be important. Those who can't, you can text documents to them and you can work with them. So let me get back over there. So we're going to go to our class today. First thing I'm going to do is I'm going to go to content. I'm going to look at today's lesson. Our unit is genetics. We're still in genetics. We are going to be asynchronous. There is a test retake next class. These are the directions to do that. That will be Friday. You'll need to go there. In all likelihood, today's exit ticket, I'm going to drop in there as well. I will take the exit ticket to on Friday, probably if we can't get to it today because of the delay, as attendance. So let's see whether we can get this all done today though. So I'm going to drop in and look at our agenda today. So we did the do now. We're about 4 minutes off already. That's not too bad. We're going to do introduction to new material. We're going to be looking at rules and facts. That's it. That's really the topic. So you can see here, these are facts and rules. These are things that you kind of use as a guidepost that you can, if you're lost, you can look up, you can see, and I'm using this figuratively, you can look to see what facts are around to figure things out. Our do now, I knew a fact. If I have a ratio that's disproportionate, like 5-to-1, then I immediately know in phenotypic ratios and a dihybrid, that's a linked gene. I answer the question, I move on and just kind of push through this. And then we're going to go into breakout rooms and we're going to have to look at some of these facts. We'll see how many of these facts that you can document. And then we're going to go over your quarter 3 projects. And you're like, oh my God. Now, last time, a lot of you guys were like, oh my God, it was a formative. This time it's a summative. This is a summative. We're going to have some time to go over the rubric and what the task is that you have. And then finally, the next ticket for today. So I'm going to go ahead and start. We'll start here, facts and rules. Okay. Well, I say I'm going to start. There we go. No, that's not what I want. All right, Randy, I need to see if you can see what I see. You're not. You're still. Sorry, I'm sharing the wrong thing. I'm looking at Mosati's screen. It did not come up the way I needed it to have. Technical difficulties, they span. Okay, time me 15 minutes. What are you going to tell me to do, Mosati, in 15 minutes? You can tell me. You can say however you want. Stop talking. Okay, so again, we know the code prior to the 1950s. It was already known that DNA was probably the code. They really kind of solidified that with the Hershey-Chase experiment. The next question was, how was this molecule put together? How was this molecule put together? And rather than just preaching to you all day, I want to give you guys a chance to look for patterns. So what pattern do you see? And if you aren't maximizing your screen, you probably want to maximize your screen so you can see some of these images more clearly. What patterns do you see? Do not answer for 30 seconds. Just see if you can see a pattern. Counting on you to keep me on time. You're keeping me on time, yes? Mosati? Abuelito made it, yes. All right, patterns. What do you guys see? Do you see anything that resembles a pattern? It's a very small strand of one side of DNA, but maybe you guys have noticed a pattern so far. Yes, there's one. Good job, Kimberly. You've got one of 2 patterns. Every sugar has a nitrogenous base. Oof. H is connected onto the N. H is connected to the N. Yes, H is connected to the N. So this is going to be my sugar. And so hydrogen is connected to a nitrogen. That's always true. So these are my nitrogenous bases. They're always connected to a sugar. There is one more pattern I'm looking for. They haven't all been found. There is another pattern. And I'd rather you look for the pattern and discover it yourself. That way you're not just being talked to. There's another pattern here. We're really looking for 2 patterns that you're going to see. The nitrogenous bases are always connected to the sugar. There's still another pattern. Not quite. And then the phosphate groups are here. They're not connected to the nitrogenous bases. It's a sugar. Yeah, it's a sugar. It's a pentose sugar. So here, yeah, there you go. So yes, Manas just saw it as well. Caleb beat you to it, Manas, that you have alternating phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar. The backbone repeats itself. Therefore, that is not the code. I would always get the same message. It would be like Morse code and sending the same code every time. The code really lies over here, but the backbone is going to be repeating. So you're going to have, this will be, who knows what this is going to actually end up being, what the pattern, there will be no pattern necessarily here. This is going to be like letters in a book, and they're not going to repeat necessarily. Where these is always going to be, think about the binding of the book. The bindings of the book are always the same. The words in the book are going to alternate, but the binding is always going to be the same. It's that corner of the book where the pages are stuck to it. Kind of think of it in that manner. And these are just factoids that you have to know about the structure of DNA. Yeah, we're going to get to the 5 and 3 does mean something, and I'm going to get to that in just a moment. Thank you for bringing that up, George. So the next thing that we're going to bring up is Chargaff's rule. So Chargaff has analyzed DNA, and he came up and he saw a pattern as well. And we're going to look for patterns. And what he did is he took DNA from all sorts of organisms. And I say all sorts, I mean, he did hundreds of species, and he found a pattern. So let's take a look at this. So when you look at this, do you see a pattern? And if you do see a pattern, fill in these boxes, fill in these on the data table. You should start to see a pattern. I'm not going to put up the questions just yet. I want you just to look at the data table. I want to really focus on data analysis on this lesson. What patterns can we see? We are pattern-seeking organisms. That is how we work. I'll put up the first question in 11 seconds. Right now, you're just looking for a pattern. Okay, first question. What is the percentage of thymine in the salmon? It's a pretty straightforward question. Drop it in the chat. Just so I know that you guys are reading it correctly. You are correct. You are correct. You are correct. Excellent. You see it right there. There's thymine, cytosine, guanine, adenine. Now, in humans, what is the percentages of guanine and cytosine? In humans, what is the percentage of guanine and cytosine in here? What would be the percentages? No, you're supposed to fill it in. Infer what goes in there. Using the pattern, infer. Good job, Keaton. Keep it to yourself. Good job, Monis. Keep it to yourself. Try again, LeVar. Try again, Jacob. I'll hold on another moment. Try again. Good job, Santos. I'm going to wait till I get 5 people to see it. Other people keep studying it, keep trying. Don't stop. Keep looking at it and thinking about it. What could it be? There's a pattern. Good job, Randy. Need one more. Not quite, George. Looking for one more. Okay, good job. So I got 2 more that did it. Okay, so let's take a look. How does this work? How did they figure this out? And I don't remember who the last persons were. I just saw numbers come up. Let's see how we could actually figure this out. So if I look at my pattern here, I've got all of these you'll see are approximately, the sum are going to be approximately the same. 32%, 32%, right? And then 17 and 17. So some of these actually match one another. Let me see if I can change. Yeah, some, and I'm going to try to bring this down here. It doesn't work. Okay, I think this is the guanine column, and I think this is a cytosine column. I can't see it because I have an annotation tool in my way. This is thymine, and this is adenine. So he made an inference. It looks like A and T are approximately the same. So let's just work on the. Let's go down to human. So I've got 30% here, 30% here. So adenine and equals 30, approximately 30%. I'm just going to say approximately. And humans are going, I mean, excuse me, humans and thymine is going to be approximately 30%. Well, I'm working on 100%, right? So 30 and 30 together plus 30 is going to equal 60. So I would just now go 100 minus 60. I've got to have 100% of that DNA, and that's going to give me 40. Now there's two more here, right? There's two more. And so I need to take 40 divided by 2, and I'm going to get 20%. So this should be approximately 20%. 20%. And if this is 29, this means that this is going to be approximately 29. And if this is 29, that means each of these is going to be 21. They all are going to add up to 100. You're going to have a total here. They're going to come up to 100. And what was so cool about this is that he saw that these approximations, if you look at this, approximately, no matter which species it was, you were close to 30% of your DNA was in adenine and thymine, no matter what organism you were. You started to get a little different right here in the bacteria. This is the one spot where we start to see a little bit of an exception. Bacteria tend to be even, relatively even for all of these, about 25% each. But all eukaryotes, everything that's a eukaryote, tends to be 30, 20, 20, 30, approximately. And this was a pattern that he noted, and no one could literally look at the molecule and see it. They just had to detect it and then get a signal and measure those signals. Again, this happened a long time ago, and they did what they could with the technology they had. So this is what we call Chargaff's rule. This is Chargaff's rule, which says that adenine and thymine are going to be equal to one another. It's our base pairing rule. And these are facts. Adenine and thymine are equal to one another, and guanine and cytosine are going to be equal to one another when you're doing this. Now, the thing about IB that they're going to make it's going to be a little different. They expect you not just to know the rule, but who actually developed the rule. So you need to have this person's name down. So those of you guys that I see in here, rather than just looking at your screen, probably you want to write down Chargaff's rule and then jot that information there. Otherwise, it's going to come back and bite you in the tail. It's going to be something that you don't want to get, you can potentially get caught not, getting correct. It's too bad. Okay, so now scientists are convinced DNA is the molecule for heredity. Let's start looking at the structure of it. Along came a spider to sit down beside her, and that's Rosalind Franklin. She's the one who figured out that it was actually a double helix. She and a couple of other gentlemen, Watson and Crick. But even Watson and Crick were having a very difficult time with it. And she says, well, I think that whenever I can figure out your problem, they couldn't figure out which direction the nitrogen bases were facing. She said that they were facing inward. And she did that by taking an X-ray and noting what she thought inferred was the double helix. At that point, they did conclude that there was a double helix. Do you notice any more patterns? Do you notice any more patterns? Patterns are where it's going to be. We got to get these facts and rules. And to get these facts and rules, we should look for some patterns. Patterns should emerge with facts. They should stay the same. What do all of the nitrogen bases have between them? What do you notice is between each nitrogen base? Okay, you guys are seeing it. Cytosine connects to guanine and adenine connects to thymine. Excellent. What is the nitrogen bases are inside the helix? Yes. What do you notice between the bases? What do you notice between the bases? There is a link. Do you have any idea what that link would be? Oh my God, you got it, Gavin. Gavin, go ahead and put it out to everybody. Drop it out to everybody in the chat, please. Gavin, go ahead and drop that information out to everybody. There's a hydrogen bond. Thank you. There's a hydrogen bond between every nitrogen base. Those three little dots, remember my hydrogen bonds between water molecules. These little dots represent hydrogen bonds. That's another rule. That's how they're stuck together. It's another thing to know. We know, again, the nitrogen bases are going to be paired, adenine, thymine, cytosine, guanine. Randy, you dropped that one in there. Excellent. But we also noticed this hydrogen bond. So how many hydrogen bonds between thymine and adenine? And I'm going to ask very specific people this. So take a look at the pattern. Abuelito, how many between adenine and thymine? How many hydrogen bonds between adenine and thymine? No, only Abuelito. Morales. I'm asking Morales. Don't put it out there, I'll hurt you. Okay, yeah, I see that. Good job, Caleb. Between adenine and thymine, how many? Okay. And I knew you were going to give me one or two answers. You were going to say 6 or you're going to say 2. What these are, that's my 2-minute warning. That's what I got for 2 minutes. Is it you tell me 15 minutes? All right, I'll hush after this one. There's going to, you would say 6 because every little dot would you think represents a hydrogen bond, but that line of dots is 1. So it's 1, 2. So well, those little circles on each side of them are going to be actually a different molecule or a different, not molecule, but a different atom that they're attracted to. So oxygen and hydrogen are attracted to each other. That's where we get these hydrogen bonds. So I don't remember which one's actually oxygen or hydrogen over here. Nevertheless, you can see that there's going to be 2 between adenine and thymine, and guanine and cytosine are going to be 3. And this makes sure that it usually prevents there from being mutations. Usually, most of the time, far majority of the time, but sometimes there's going to be a problem. Here, it's a little more illustrated. You can see what atoms there are. You see hydrogen and oxygen, or in this also here, you see nitrogen and hydrogen also have these hydrogen bonds. Last question before I go to the guided practice, and don't answer out loud. I want you to think about it and drop it into the chat. Are hydrogen bonds strong or are they weak? And in 5 seconds, I want you to drop it into the box. This will be important later. Drop it into the chat. Are hydrogen bonds strong or weak? 3, 2, 1, go. Are hydrogen bonds strong or weak? Yes, it's not an answer to that question. It's about a mix of 50-50. Okay, it is a real mix. All right, although we are getting more weak than strong, and it is true, it's weak. If they were strong, you would not be able to boil water. You would not be able to take a bath. You would not be able to drink water. Water molecules, it would mean water was a solid. It's usually, at room temperature, not usually, but at room temperature, it's really weak, right? You can get in it, take a bath, you don't break your back trying to get into water. Hydrogen bonds are weak bonds. Water can evaporate more quickly because these bonds separate very, very easily. So these bonds that put DNA together in the middle, whoo, you talk about easy to break. That's important. That was really important for Griffith's experiment where he worked with the mice for the pneumonia. You could break this and it could reassemble itself very, very easily. It reassembles itself. So when he could denature the proteins and they go back to the wrong shape, but you can heat up DNA, it can fall apart and reassemble itself right to the same pattern it was before. They're very weak bonds. All right, I'm going to go to Schoology. That's where everybody wants to be. I want to go to Schoology. Yeah, I do. Oh, yeah. I'm going to facts and rules. Facts and rules, first things first. Before we go to our model, we look at our model, we're going to go to guided practice. There's a couple of you guys sitting in the chat. So what's the, oh, it's weak. It's super weak. That's the answer. I mean, if it was strong, you wouldn't be able to take a shower in it. It would be like taking a shower with rocks. So water breaks super easily because it's held together with hydrogen bonds. You can jump into a pond and not shatter into a million pieces. You can break yourself. It's a very weak bond, very weak bond. All right, so let's see. I'm going to go to DNA guided practice, open this document. And it's full of rules. Many of them we have already learned today, and then some that we learned the other day. I want to see how many of these you guys can get filled out in 15 minutes. 15 minutes. And here we go. I'm going to drop you into breakout rooms. If you are not able to get into Schoology for whatever reason, then you can have somebody make a copy of the document and send it to you. Get ready. Go. It didn't matter. Y'all could. I did not make it. I wanted you guys to have this hard copy saved, and then I'm going to print y'all and give them to you. This one's pretty easy. I can drop y'all all in the same room. Y'all want me to put y'all in the same room? All the ugly kids in the same room. I can literally, oh, I can't add a room, but I can make an ugly kid room. I can do where I just drop all the ugly kids in there. Right now we've got Abel, yeah, like Caleb, you and Masadi are in the same room already. Uh-oh, hold on a second. Somebody's texted me. Sorry, you can write on the document, or y'all can't. Shoot. All right, who is in your room? All right, so you send them your email, they'll send you a copy of the document. It's just that easy. Come on, there's workarounds here. You say you had pride. Let's do it. Show some. Watson, are you not able to get there? Daniel, I saw your, I cannot send a Zoom link. If I cannot send a Zoom link, I don't think. Let me see. Maybe I can. Let me see.
Wait, Mr. Pope, can you send it again?
What, the
Is it
Zoom link?
like to join the breakout rooms?
Who is Watson? You should. It shows that you should be able to join a breakout room right now. If I could drop you into another one. Hold on. How about this one? Look and see if you can get in here. Do you see that?
No, it didn't pop up.
Really? I'm just dropping. I can drop you back to the original room that you were in. Look around the edges. I don't know why it wouldn't pop up. I'm dropping you back and forth. Expand your screen to be big, like full screen. I'll go in there and get him. Yeah, he's in there. I can see he's working. No, I doubt it. I'm about to go in there and find out. I doubt it.
Wait, I'm like, wait.
Can you?
Mr. Pope, it kicked me out.
Okay, I'm gonna try it one more time. And you went in and it kicked you out? Try that one. You have me on email, send me an email, and I will drop the document to you. It won't open on mine. If you,
said you sent it.
all right, check your email. Where's Daniel?
Just like a true or false.
Daniel, I got your email.
Yeah.
I'm gonna send you the link. Can you send it to Amarani?
Yeah. Yeah.
All right, I'm gonna text this. That's the Zoom link. Everybody can see it. Send it to Amarani.
Okay, we'll do.
Okay, let's check out everybody's progress so far. Oh, wrong class. Okay. Facts and rules. Document. DNA guided practice. Let's see. So who's in here? Let's see. LeVar. Are you guys sharing your screen? Or did I even give you guys permission to share a screen? Yeah, you guys can share your screen and work your way through them. It'd be a lot easier if you guys talk about them, that's for sure. Buckman, here you are. Buckman's in here.
Oxygen. Hydrogen. Hydroxide.
Which
Oxygen hydroxide.
which number are you on?
Huh?
Which number are you on? I haven't got there yet. There's some of you're gonna be able to do, and some of them you're not gonna be able to do.
What do you call it when you put hydrogen? Monoxide, right?
What do you call it when you stay on track and do what you're supposed to do in class? What do you call that?
Academic honesty.
No, it's called pride. You said that you're gonna keep some. Keep some. Let's jump into this group. You guys talking through this, or you being quiet? Yeah, all your microphones are on. Why would you do that? I don't understand. You guys will get a lot further in less time if you talk. Just a sneaking suspicion.
Oh, we have a shared document that we're all editing.
Yeah.
And you are working on a shared document. Do you mind showing me? Come on, y'all can't be almost done with the entire thing. Come on, show me.
It's in the chat if you
Oh, it is?
But you can't
Yes.
see it.
Drop it in the chat again.
Okay.
You're not going to be able to answer every one of these yet, at least unless you're just looking them up. Yeah, some of these you guys are already, you're just looking them up. That's fine to look them up. You're working ahead. Wow, you guys are almost done. The one thing you're not going to be able to do is label this. That'll be up to you to draw and label yourself. Yeah, you're way ahead. You guys are way ahead. How are you guys finding all these answers? Are you just Googling the answers?
A lot of it was in the reading for the text.
Yeah, it was in the reading too. Yeah, I know it was in the reading too. I get it. All right, that's 1 minute.
If you're trying to talk right now, you're muted.
Yeah, somebody asked about the capsule and the bacteria, and I was just explaining that on the Griffiths experiment, the smooth strain had a capsule, while the R strain did not. That was in the reading as well. I went to two groups, and two groups were explaining to me that they were able to work ahead because they did the reading. The reading is really important. Patty said she still has books in the office. She hasn't taken them back. So if you haven't picked up a book yet, I don't know how many people at home have not yet. You might want to get that, especially in this unit. It's going to get pretty tough. We're going to forego the exit ticket. The exit ticket is going to be for Friday. I want to make sure that we have a time to go over our rubric. I'm going to open the rubric for our quarter project so that you are very clear as to what we're going to do. So I'm going to open this document. And again, this will be a summative. You can see what the due date is going to be as well. The due date is going to be what? Caleb, what's the due date? You don't look at me, look on the screen, you can probably see it. Due date for the project.
March 1st.
That's right, March 1st. That's approximately how many weeks?
That's 2 weeks.
Nope, it's about 3. It's about 3. Okay, 21 days is 3 weeks, just heads up. Okay, so we've got, you're going to build a model. Now, the kids that are in this room, you're going to build a physical model. You can see sort of examples of them hanging in here. You can see some sort of examples in this room. Some of these are appropriate and some of these are not appropriate. I want you guys to look at this rubric and then ask me some questions. I'll give you guys 5 minutes to open the rubric and look at it carefully. You want to expand your screen. If you're looking at it on my, through my Zoom, that's a mistake. You want to open it yourself. You want to open it yourself. If you cannot get to it, I'm going to send you a copy of it in the chat. You need to open this and then make your own copy. I'm going to freeze this so it cannot be altered. This document, yeah, it's viewer only, so you can just look at this one. I sent you the document. I'll give you 5 minutes. No, in there. I need that turned upside down. That phone, you can go away. You're wasting your time. You're not gonna waste it in here. You're looking for some things like I'm not sure about. Could you give me more clarity about? The last part of the rubric, the questions, don't worry about that yet. I haven't shared it with you. Again, if you're in person, you're going to have a physical structure. You might want to jot down some questions that you can field to everyone. Got 2 more minutes. Okay, I am open for questions. Please don't chat them to me. if they're chatted to me, chat them to open to everyone so that you can have other people read them to me.
If we're virtual, how exactly do we share? Like, are we presenting these?
Yeah, you're going to present them. It's going to
Okay.
be, it's going to be high stakes. Like
How do
high
we
stakes.
Oh,
So what you would do is you would put it on, for me, if I were going to do it, I would put, you can use whatever program you want. You could potentially use a video, you could use a PowerPoint slide, but you've got to use something where you're going to make kind of like a collage. It's going to have to be something representative of each, like for instance, every nitrogenous base, guanine needs to be the same repeating figure. Don't use like a literal representation of a guanine. Maybe use something else that could represent guanine and make a piece of art. And you could just assemble it on a slide, like or you could, maybe some people have programs where they can actually draw, and then when they draw whatever they're going to use to represent that, maybe it's going to be, I don't know, I can't imagine, maybe a battery. I don't know why you'd use a battery. I have a battery sitting here, and then they have that same repeating battery. They can make copies of whatever they actually drew. I want it to be kind of cool and pretty, like really beautiful. The kids in the class, they got to make a physical hard copy of this, like a physical structure. Does that answer your question?
yes, it does. Thank you.
Okay. Other questions? And if there are questions, they need to be audible. If they're in the chat, chat them to somebody else that can read them to me if you cannot use your microphone.
LeVar asked, will you be asking the questions or will other students be asking them?
Yeah, I'm about to share the questions with you in a few minutes, what they're going to be. I'll be asking the questions. I have a bank of questions. I have a jar with little beads in it, and every one of them has a number on it, and I'm going to randomly pull out 4, and you'll be responsible for whatever 4 questions I pull out of a jar. And I'll show you in a moment. What other questions do you guys have?
What do you mean by structure functions? Like, or like, how do we show it if we're doing like virtually?
It will virtually, you're going to share your screen and you're going to project that image for us, the entire class to use. And you'll be able to use your mouse to kind of explain, oh, this is this and this is that. This is why I made it this way. So here's my these represents my phosphates. These represent my hydrogen bonds, either the covalent bonds between the phosphate group and the sugar. This is what I use for each of those. Here's my 5N, here's my 3N. This is how I signify the 5N and 3N. You can do it however you want, but you just got to project it. It could be a still image. You can have it where it replicates itself and it's a moving image like a GIF or a GIF or whatever the hell that thing is called. What I really want to get down to, though, for the people that are here, when I get to this part for atypical stress, the kids that are in this class, atypical stress is non-typical stress. I need to be able to take your project and throw it and it not fall apart. To get the full credit here, I need to be able to take that sucker, throw it against a wall and it not shatter. If it does, no dice. So like if it just falls apart because you're having to hold it and it's just barely holding together, that's complete failure, right? If it deteriorates under typical stress, that's just like us taking it and just picking it up. If it holds up and it doesn't fall apart, but maybe breaks a little bit, that could be that would get you 3 out of these 4 marks. And then here to get this full credit, to get really the 100% is where it doesn't break. And so if you look around the room, the kids that are in here, you can see some of these materials are really, they didn't do very well. And some of them are good and solid, really hardcore. Some of them, like those back there, champions. I could take those things and toss those things across the room and they're not going to break. Most of the kids that like theirs the most take them home at the end of the year, but they have to have all these scientific principles. So these are, you might want to come in here and take a look. You can see like that one fell apart straight out of Locash. That one just trash right there. That one that's only got one side to it, it fell apart just hanging. It just kind of depends. You can always tell who puts in a lot of work and effort into this. I'm gonna go to the questions real quick.
We have another question in the chat. So
All right.
Kimberly asked if even if we are virtual, can we make an actual structure, like a hard copy
And bring
structure?
it and come to school and present it that day?
No, she just means like, can she share it on her camera, like from her house?
It's gonna have to be if you're virtual, it needs to be virtual. Otherwise, we won't be, it's looking at it through a video, it's not gonna be good. It's not gonna be terrible. Sorry. The kids in here are probably thinking they wish they had a virtual one. Well, that's the wrong question, sorry. Let me go to the questions, open up the wrong document. Yep,
How exactly do you want us to do it if we're virtual?
you're just gonna make a virtual copy, like an image, like artwork, like a collage, like a really pretty picture of a DNA molecule. That's it. Like you could put it on one slide, or you could put it on a video where you're assembling things. There's a lot of different ways that you could potentially do it. It just cannot be, it needs to be something that represents a phosphate group. It can't be like, you don't want to have something that is a phosphate group, but you can see the pattern because you're using the same, whatever image you're using for a phosphate group. Say it's going to be a ball, you're using that same ball for that phosphate group every time. You can look online like for collages and you can see it. Just we need to be able to see those patterns. I do want to get to the questions though, so that you guys are clear on these questions, and you can open that document as well. Look at think of some of these questions you can answer already. So if you're looking at these questions right here, we can already see, like we should be able to answer this one today. We should be able to answer this one today. On that day, there are 27 questions. 27 questions. I'm gonna have 27 beads in a jar, and I'm gonna shake that jar. And I'm gonna say, Abuelito, 1, 2, 3, 4. I'm gonna take out 4 beads. And if I say, question number 27, is DNA replication conservative, semi-conservative, or dispersive? You need to tell me the answer. And you don't know which one I'm going to pull out. And that's 25% of your grade. However, every one of these questions comes up on your exam. So why wouldn't I not? You need to memorize all this.
Mr. Pope, someone asked if the project's individual.
Yes, it's individual. And it is a summative. That's why you're getting 3 weeks for this. It's not that taxing, to be honest. I've had, I traditionally give people 2 weeks to do this, but we're, in a shutdown. You guys have a 4-day weekend this weekend, essentially. I know you go to class on Friday, but it's probably not gonna be that taxing. So it's a good weekend to kind of get started, assemble a plan. I have had students that get together in groups and talk about what they're going to do and how they're going to do it, and they make similar copies of it, but it's really not that big of it. That tough as long as you start early. And when I taught freshmen, I did the same project with freshmen as well as juniors. So all of them have had this. So I know you guys can handle this for sure. Other questions? Like there are 3 components.
If we're virtual, we can present it either by like for example, like me talking right now while I show a picture, or I could show you a pre-recorded video.
You could do either, but you'll have to give us a pre-recorded video in class.
Okay.
Like we're gonna go kid to kid to kid. It's gonna take 2 days to do it. So we'll do it on the 1st and on the 3rd. And I'll randomly choose who's gonna go that day as well. I've got a little, it's called popsicle sticks, and I'll go with my roll on there, and it'll be bloop. It'll be, okay, it's Diana Santos. She's the first one. It'll be completely random who goes when.
If we're doing it virtually, we can use like any symbols we want, right?
Any symbol you want, as long as it's not something that's obscene. So don't use genitalia as a nitrogen base. I won't be happy with that. I'm sorry? It's due on the 1st. It's on that rubric. It's due on the 1st. All right, I'm gonna leave you guys be. You guys play in the street. I will see you guys. Oh my gosh, I'm not gonna see you till next week, but make sure you come in and you handle the exit ticket. It's really subjective. It's the easiest grade you're gonna get on Friday. You guys be good. I'll clean your all's desk. I'll clean your all's desk.
Mr. Pope,
Yep.
can we work in groups?
Everybody has to have their own project, but you can work in a group to get it done. But everybody has to have their own one.
Bye.
You'll never forget this information. It'll be the easiest thing you have on the test after you build it. I'll answer questions while I'm cleaning desk. If you guys don't have questions, I can hear you. I'm just cleaning desk in here. If not, you can go to your other class. What's up, kiddo? Those are plain. How's your day? Oh, man, it was actually better than I thought it was gonna be when I saw class length was now. Kids get a workaround pretty quick and easy. Yeah. Kind of gave me a bit of a fright. So did you guys get a workaround in your classes too?
Yeah, I mean, it was working for me.
Oh, was it?